combination of MEG and fMRI to map the spatiotemporal human brain dynamics of a visual image being recognized. For nearly a decade, a team of MIT Computer Science and Artificial Intelligence Laboratory researchers have been seeking to uncover why certain images persist in a people's minds, while many others fade. To do this, they set out to map the spatiotemporal brain dynamics involved in recognizing a visual image. And now, for the first time, scientists harness the combined strengths of magnetoencephalography, MEG, which captures the timing of brain activity, and functional magnetic resonance imaging, fMRI, which identifies active brain regions, to precisely determine when and where the brain processes a memorable image. Their open-access study, published this month in PLOS Biology, used 78 pairs of images matched for the same concept, but differing in their memorability scores. One was highly memorable and the other was easy to forget. These images were shown to 15 subjects with scenes of skateboarding, animals in various environments, everyday objects like cups and chairs, natural landscapes like forests and beaches, urban scenes of streets and buildings, and faces displaying different expressions. What they found was that a more distributed network of brain regions than previously thought are actively involved in the encoding and retention processes that underpin memorability. People tend to remember some images better than others, even when they are conceptually similar, like different scenes of a person's skateboarding. We've identified a brain signature of visual memorability that emerges around 300 milliseconds after seeing an image, involving areas across the ventral occipital cortex and temporal cortex, which processes information like color perception and object recognition. This signature indicates that highly memorable images prompt stronger and more sustained brain responses, especially in regions like the early visual cortex, which we previously underestimated in memory processing. While highly memorable images maintain a higher and more sustained response, for about half a second, the response to less memorable images quickly diminishes. This insight could redefine our understanding of how memories form and persist. The MEV-fMRI fusion method, developed in the lab of CSAIL senior research scientist Aude Oliva, adeptly captures the brain's spatial and temporal dynamics, overcoming the traditional constraints of either spatial or temporal specificity. The fusion method had a little help from its machine learning friend to better examine and compare the brain's activity when looking at various images. They created a representational matrix, which is like a detailed chart, showing how similar neural responses are in various brain regions. Picking the conceptually similar image pairs with high and low memorability scores was the crucial ingredient to unlocking these insights into memorability. Lahner explained the process of aggregating behavioral data to assign memorability scores to images, where they curated a diverse set of high and low memorability images with balanced representation across different visual categories. Despite strides made, the team notes a few limitations. While this work can identify brain regions showing significant memorability effects, it cannot elucidate the region's function in how it is contributing to better encoding retrieval from memory. Understanding the neural underpinnings of memorability opens up exciting avenues for clinical advancements, particularly in diagnosing and treating memory-related disorders early on. The specific brain signatures we've identified for memorability could lead to early biomarkers for Alzheimer's disease and other dementias. These findings are exciting because they give us insight into what is happening in the brain between seeing something and saving it into memory. The researchers here are picking up on a cortical signal that reflects what's important to remember and what can be forgotten early on. Lahner and Oliva, who is also the Director of Strategic Industry Engagement at the MIT Schwarzman College of Computing, MIT Director of the MIT IBM Watson AI Lab, and CSAIL Principal Investigator, join Western University Assistant Professor Yalda Mohsenzadeh and York University researcher Caitlin Mullen on the paper. The team acknowledges a shared instrument grant from the National Institutes of Health, and their work was funded by the Vannevar Bush Faculty Fellowship via an Office of Naval Research grant, a National Science Foundation Award, Multidisciplinary University Research Initiative Award via an Army Research Office grant, and the EECS MathWorks Fellowship. Their paper is published in PLOS Biology.